Hi everyone, my name is Mohamed Babi. I'm an imaging specialist at the Center for Advanced Light Microscopy. We're a core facility in McMaster University here in Ontario, Canada. And today I want to give you an overview for sort of the different optical microscopy techniques that you can use in your research and also highlight some of their applications. Now in the presentation, I'm mainly going to be going over different microscopes that we have in the facility and some of the applications that we've been using them in, but the takeaway message here is that all of these techniques are actually the same in the working principle. And so although the microscopes will actually be different, they're all working through the same sort of fashion. And so I'm going to break down the different optical microscopy techniques, starting off with the most simple one that we might have all seen before, which is called wide field microscopy. And this is typically what we think of when we're thinking of using an optical microscope, like the one on the right-hand side. It essentially consists of a light source, either from the bottom, if you're using an upright microscope and doing transmitted by light, or from the top, again for an upright microscope if you're doing fluorescence imaging. And upright microscopes, inverted microscopes mean whether we're actually looking at the sample from the bottom or the top. So in this upright microscope here, we're looking at the sample from the top, and we could use transmitted white light or bright field to look at our samples very quickly. The wide field microscope uses a camera to capture your image, and because cameras are very fast, we're able to quickly capture images, and we're able to look at all sorts of different samples very quickly. The nice thing about bright field or transmitted white light imaging is that we don't necessarily need to label our samples with anything. So we could look at, for example, cell morphology, cell division, and dynamics without having to do any sort of labeling like what we see here with the transmitted white light image. We can also use different contrasting methods like bright field, differential interference contrast or phase contrast or polarization microscopy to get a little bit better contrast, or even look at, for example, crystalline materials and amorphous materials if we're using polarization optical microscopy. So wide field microscopy can also be used to do fluorescence imaging, and fluorescence is usually required if we want to look at more specific cellular targets, if we want to visualize some subcellular organelles or look at the localization of different proteins. We need to be able to fluorescently label these specific targets, usually using an antibody that's conjugated to a fluorescent probe. And because wide field microscopy is so fast, now we're able to image many samples in a short period of time. And this is typically done with something like a plate reader. On the left-hand side, we have an example of EVOS plate reader, which is a very popular instrument that cell biologists use. And these plate readers usually have molarized stages, and so they allow us to automatically scan different samples in a multi-well plate. And they often also support live cell incubation, which allows us to do time-lapse imaging. So with these kinds of microscopes, you can gather lots of data very easily. Imagine now you're scanning a 96-well plate, you're doing a time-lapse over 48 hours, and if your samples have multiple layers of cells, you're also taking Z-stacks. So you're collecting lots and lots of information because it's very fast in collecting those images. You can get more fancy plate readers, like for example, this Cell Discover 7, which has more fluorescence channels and more automatic focusing routines. But all of these different microscopes are all working through the same principle, which is wide field microscopy. And you can get even nicer images if you perform, for example, image deconvolution if you have thicker samples. The other flavor of a wide field microscope you'll see is a slide scanner. And these instruments typically take 20 to 100 or even more slides, and they're automatically scanning the sample by taking an image, moving to the next sort of space adjacent to that image, taking another image, and scanning this sort of sample tile by tile, and then eventually merging them all together to make a very large image. And this is typically done with tissues. And the nice thing about the slide scanner is you can get very nice sort of high resolution images at the single cell level but within the context of the entire tissue. So you usually have very large images generated this way. This, for example, has 50 by 60,000 pixels all in one image, and you're allowed to see every single cell within the tissue. And because you're scanning the whole slide, you can put multiple tissues on the slide, and you're able to load up to 100 slides to look at all sorts of samples in fluorescence or H&E stained samples with bright field. So the nice thing about wide field again is that you're able to image lots of samples very quickly, but there's always going to be some pros and cons to every microscopy technique that you're going to use. So if you're imaging very thin samples, for example, you're plating cells on multiple plate or glass cover slip, a wide field microscope is going to be giving you a beautiful image like the one we have on the left here of a plated fibroblast. But what if you have a much thicker sample? Say you're looking at a 5 micron or 20 micron thick piece of tissue, which now has multiple layers of cells within the tissue. What you're going to see is you're going to see a lot of background in the images because you have out of focus light coming from the top or bottom section of your sample, and that's entering the detector in the form of background light. So you won't get a lot of contrast. And this is where a different microscopy technique is needed. Usually confocal microscopy would be used to block this out of focus light and give you much higher contrast in your image, so now you can actually see the individual layers within your tissue or thicker sample. Now, does that mean that you always need to use a confocal microscope? No, because if your sample is, again, these very sort of thin cells that have been plated on a multiple plate, then you don't necessarily need the ability to block out of focus light because you have just one plane that you're going to be imaging. So on the left-hand side here, we could see that the confocal microscope is going to give you just as good images as the wide field microscope, and the wide field microscope is arguably going to be better because the camera actually has less noise than the detectors used in the confocal microscope. And you can get all sorts of different confocal microscopes that can have upright and inverted configurations, and they can have different bells and whistles on them. But typically speaking, a confocal microscope will have 3 or 4 different fluorescence channels that you can use to image your sample. And there's going to be different kinds of confocal microscopy techniques like line scanning microscopy and spinning disk confocal microscopy. And these are all, again, working through a similar principle in that they are blocking out of focus light to give you nice optical sections and high contrast. But they all have their own advantages and disadvantages. And the choice of the microscope should really be driven by the kind of sample you have and the research question that you are trying to answer. And so now I'm going to go into a little bit more technical detail about what a wide field microscope is and how it's different from a confocal microscope. So again, a wide field microscope is just a regular optical microscope when we think of using a microscope in the lab. And for a fluorescence wide field microscope, we usually have some sort of light source here that's producing white light. But when we're doing fluorescence, we have these labels in our sample that absorb a specific color of light, and then they re-emit that light back at us in a different color. And so if we have a way of separating the light that we're exciting our sample with and only looking at the light that is actually being emitted by the sample, we get these beautiful high contrast, sort of glow-in-the-dark images, and we can use different dyes with different colors to look at multiple components independently and then again simultaneously to see the way that they're interacting. And so we need to have a way of being able to excite the sample with a specific color and then collect the emission from that sample in the other color and separate these two apart. And so this is typically done with a component called a filter cube. And the first component of the filter cube is an excitation filter, which is going to take the white light that's coming out of your light source and filter it to give you a specific color of light that we can use to excite the sample. In this case, we have, for example, a Tritzy filter cube, which has an excitation filter that allows green light to pass through. Then the second component of the filter cube is a dichroic mirror. The dichroic mirror is going to reflect that green light up. And this is an optical diagram for an inverted microscope. So the objective is looking at the sample from the bottom. And so the dichroic mirror reflects the light up, and the objective projects the light onto a large area of the sample or a wide field of the sample. And this is why it's called wide field microscopy. In this case, the sample is a cell that's been fluorescently labeled, and it's going to absorb that green light and then fluoresce and emit that light back at us in the form of yellow to orange to red light. And so the objective is going to collect that light back down and focus it onto the camera. But as the objective is focusing the light, it's going through the dichroic mirror. So the dichroic mirror is special in that it will reflect green light up, but allow all the other wavelengths of light that are longer than green to actually pass through. And so now we're able to separate our excitation light from our emission light. But then we need to further clean up the emission light because it's still a spectrum, and we want to look at a specific part of that spectrum. So then the third component of the filter cube would be the emission filter, and that's going to take the yellow to red emission light and only allow us to look at the orange light. And again, now the image is focused onto a camera, which has a sensor with thousands of tiny pixels that will be able to detect light from different parts of the sample simultaneously. So this is what makes wide field microscopy very fast, and that we're able to look at different parts of the sample almost simultaneously and able to generate an image very quickly. And this is how our phone cameras work, for example, all through the wide field principle. But you'll notice that when we're using a wide field microscope, we're flooding the sample with light. So we're illuminating all the different layers of the sample with the excitation light, and we're getting fluorescence from all of these different layers. Even though our objective is focused on a specific layer, it's actually still collecting the light from maybe the top or bottom portion of the sample. And that's going to give us lots of background light if the sample is thick enough. And this is where a confocal microscope would be used, which has lots of sort of differences from a wide field microscope. Usually in a confocal, specifically for a line scanning confocal, we have a laser which already has a very well-defined wavelength or color, so we don't need an excitation filter. Now we have an optical diagram from an upright microscope, so the objective is looking at the sample from the top, and then the laser is going to be shined on the dichroic mirror, which is going to reflect the green light, in this example, down. Instead of illuminating a wide field of the sample, the confocal microscope is going to take the laser and focus it onto a very small point, a very small volume, so that most of our illumination is actually focused on one focal plane. But you'll notice that we're still having light pass through the top of the cell and the bottom of the cell. So it will still have different layers of the sample be excited. But with the confocal microscope, when we're collecting the light back up, we focus the emission through the dichroic mirror and then through a pinhole. And this is what makes the confocal microscope very good in blocking out of focus light, because only light that's actually coming from your focal plane will be able to pass through the pinhole and hit the detector. And the detector is not going to be a camera. It's usually a photomultiplier tube. And we can think of that to just be one big pixel. And so we're not looking at different sort of parts of the sample. We're only looking at that one point that we excited. So then how do we generate an image? We take that point and we scan it along the line. We drop down, we scan another line, and we do this line by line sort of raster scanning to build our two-dimensional image. Because the pinhole is allowing us to look at just one focal plane, we can change focus and capture all the different sort of layers within the sample and combine those to make a nice three-dimensional rendering or representation of the sample itself. But as you can imagine, because we're building the image pixel by pixel in line scanning confocal microscope, it's much slower than wide field microscope. So if your sample is not thick, or if you don't need that high amount of axial or Z resolution, it's better to use a wide field microscope because usually it's more gentle on the sample in terms of how much light you need to put on the sample, and you're able to take your images much faster. But confocal microscope makes beautiful high resolution 3D representations of samples from single cell subcellular level. For example, here we have a T cell or red blood cell that's incorporated with cancer cell, and we can see this interaction very clearly in images. We can take confocal microscope and use it to large structures, for example, these organisms that have intelligent on gel. And if you have confocal confocal or resonance confocal, can acquire these large volume data sets in high resolution relatively. And typically, you can do this with up to 4 different colors, but some microscopes can also have up to 7 or 8 different lasers with very tunable detection ranges that allow you to look at any 4 or up to 7 or 8 to do spectral mixing. So we've seen a lot of applications, these techniques to research in biology, but optical microscopy can also be used to look at samples in the material science. For example, this here is a sample of lipid nanoparticles carrying the Pfizer COVID vaccine. These samples have been embedded in the film, and we can use the confocal microscope to take a subset through that film and visualize the differential distribution of these nanoparticles across the film. And for material science in particular, the upright microscope is going to become more useful because now we can look at the top surface of these materials that might not be completely transparent, or maybe there's only cells on top of the actual scaffold or 3D printed structure. And so in addition to operating the confocal microscope for laser scanning microscope in fluorescence mode, you can also look at the way that the laser is scattering off of the surface, and you can get some nice topographical information. For example, in this case, the confocal was used to measure the thickness of a PDMS film that's been spin coated on a glass substrate, but on only 1/2 of the substrate. So we can look at the way that the laser is actually reflecting off of the different interfaces within the sample and look at the thickness of the film based off of that PDMS to glass interface. You can also use the confocal to look at the topography and the structure of different electrodes or other surfaces that you might be fabricating in the lab. Finally, biology, you can use microscopes, such as using these water injectors, hydrogel materials with cell that is growing in three-dimensional space. And the water dipping objectives in this case are going to be very useful in that you're able to perfectly match the index of refraction of the sample to what the objective is supposed to be using. And this is a very important point that needs to be sort of addressed when you're using an optical microscope, which is to choose the right objective that will correct for the right index of refraction and the thickness of your substrate or the sample that you're looking at. So we can talk about that in more detail during the clinic. But aside from choosing the right optical microscopy technique, you also have to choose the right objective for your sample. So we had a very quick crash course on optical microscopy, and saw how wide field and confocal techniques could be used to look at entire tissue sections down to a single cell level or subcellular level in 3 dimensions and different components, especially when we're using a confocal microscope. But the resolution of an optical microscope is not infinite. There's always going to be a limit. And that limit is dictated by the diffraction of light, which is usually going to come around to 200 nanometers. But we know that biology and structures don't stop at the 200 nanometer scale. If we're looking at mammalian cells, there's smaller organelles like the centrosome or transport vesicles or molecular complexes like focal adhesions that are all occurring in this sub 200 nanometer space. And outside of mammalian cells, there's all sorts of viruses and bacteria with structures and processes that are happening below this 200 nanometer space. So if we want to study these living systems and be able to benefit from the specificity of fluorescence microscopy to study these very small particles, we need to look at different techniques within optical microscopy that can get us past this 200 nanometer limit. And the microscopy techniques that can go past this diffraction limit are called super resolution microscopies. And there's all sorts of different flavors of super resolution techniques. I'm going to quickly go over some of the main techniques involved in looking at these small particles. before we can understand how a super resolution microscope works, we need to understand why we're actually limited in resolution in the first place when it comes to a wide field or confocal microscope. As I mentioned, this limit comes from the diffraction of light. When light is passing through an objective lens, so if you have two light rays going through the objective lens, they're going to get focused onto a point. But unlike what you might have seen in the high school optical diagram, that focal point is not infinitely small. There's going to be a diffraction or interference pattern happening because of the different sort of points of light that are actually going to be emitting from the objective. So if you remember from high school, there was a single slit and a double slit experiment that's producing this diffraction pattern. And that's actually what's happening when you pass light through an objective, but it's happening in three-dimensional space to produce a diffraction pattern that's called the point spread function. And the dimensions of the point spread function are what's actually going to dictate the resolution of the optical microscope. And there's a formula for calculating these dimensions in both the lateral space, so the XY plane, and the axial space in Z. So the formula is lambda over 2NA. Lambda is the wavelength of light that you're using to look at the sample, and the NA is the numerical aperture of the objective. So the longer wavelengths and the higher the numerical aperture is, the better your resolution will be. And the axial resolution is lambda over NA squared. So your Z resolution is usually significantly worse than your lateral resolution. So what this means is that no matter how small your structures are, even if it's a single molecule or a single protein that's emitting light, if you're collecting the light from an objective lens, you're always going to produce a spot that's at least 200 nanometers in size. So you can never actually see anything smaller than that point spread function in the microscope. Unless now you're using super resolution techniques that play different sort of tricks to overcome that diffraction limit. One of those techniques is called image scanning microscopy, and a popular or commercial implementation of this is the Zeiss area scan technique, which is essentially a confocal microscope, but instead of passing the light through a pinhole and detecting it by a photomultiplier tube, we remove the pinhole, and in its place we have an array detector. So you can think of a very small camera here with tiny little pixels that each represent a very small pinhole. And if you go deep into confocal microscopy, you'll know that the smaller the pinhole is, if it's shrunken beyond this one area unit or the size of the point spread function, you get higher lateral resolution. And if you completely close it down, you can get a resolution of up to 140 nanometers. But if you completely close the pinhole, you completely lose the light that you need to use to actually collect or capture an image. So what these image scanning microscopes do is they place a small camera with very tiny pixels that each represent a closed pinhole. And each one of those pixels is detecting an image as if you were imaging with a completely closed pinhole in the confocal. But because you're not getting a lot of signal from each one of these detector units, all of those pixels are then combined together to give you a much brighter image as if you were imaging with a pinhole size of 1 or 1.25 area units. So the takeaway message for this technique is that it's essentially a confocal microscope using a different detector with some image post-processing like deconvolution and pixel reassignment to give you almost twice as much resolution, down to 100 or 120 nanometers, which is twice as good as a normal confocal microscope. And because it's based on confocal microscope, these techniques are very easy to use, and you can acquire beautiful images, for example, of different microbes like bacteria or yeast, and look at how different proteins are localizing in the membrane side of the bacteria. The other technique that can give you a twofold improvement is called structured illumination microscopy, and this is working through moire fringes. So if we imagine our sample to be essentially a pattern with a specific frequency, what we could do is we can illuminate that sample with a different pattern, and when we have two patterns over the top of each other, we get more fringes, which are much easier to see than the actual small details within the sample. So what structural illumination does is that it creates a mask and scans the sample across that mask, rotates the mask, and does it again, and keep doing that 3 or 5 times to get different moire fringes with different orientations. And if you know the frequency of the pattern that you're imaging with, and if you can see the moire pattern with the microscope, then you can back calculate the frequency or the feature that gave rise to that moire fringe, typically allowing you to double your resolution to around 100 nanometers. So the image scanning microscopy and the structural illumination microscopy techniques, they're actually quite complex in their sort of operating principles, but on application, they're much easier to execute. Usually, if you can use a confocal microscope or a wide-tool microscope, then you can apply these techniques to your samples with some small caveats on how you're preparing the sample. And they're allowing you to get almost twice as much resolution as a normal confocal or wide-field microscope. But the 100 nanometer resolution is still not going to be good enough to see very, very small things like transport vesicles or molecular complexes. So there's other techniques that can go even smaller in scale, and one of those being the stimulated emission depletion microscopy or STED. And the way of understanding STED requires us to look at this Jablonski diagram, which is the energy or electronic transition diagrams within a fluorescent molecule. So just a very quick crash course on this. Fluorescent molecule is going to have different energy levels for the electrons in that molecule. And the way that fluorescence typically works is that you excite the sample with a specific wavelength of light that will bring the molecule's electron from the ground state to the excited state. Then the electron is going to relax a little bit and lose a bit of energy when it's in the excited state, and then drop back down to the ground state. And when it drops back down to the ground state, it emits a photon with a lower energy and therefore a longer wavelength than the photon that we use to excite the sample because of that relaxation that happened at the top. And that photon is going to be your fluorescence light. And so fluorescence microscope is separating the excitation light from the fluorescence light to give us these glow-in-the-dark images. But when the electron is in the excited state, we can excite the molecule again with a different color of light that will stimulate the emission of a photon from the sample with the exact same color, if that color of light matches this exact sort of difference between the excited state and the ground state. And this is happening at a different color than the normal fluorescence process. And if we excite the sample with enough sort of illumination power using this stimulated emission laser, we can completely quench this fluorescence process. And this is what stimulated emission depletion does. You have a confocal microscope with an excitation spot, which is diffraction limited at around 200 nanometers. And on top of that, you overlay a depletion donut that completely depletes the molecules from going through the fluorescence route and instead allows them to only go through the stimulated emission route. And so if you overlay the donut with the excitation beam, only molecules within the center of the donut that are not being depleted actually emit light. And if you combine these two illumination patterns together, you go from a 200 nanometer emission volume down to a 30 nanometer emission volume. And that's essentially the resolution limit of this microscope. And you can use it just like you would with a confocal microscope, but typically you have to choose specific dyes that could be depleted properly by the donut. An example of this would be looking at the axonal cytoskeleton, which typically looks like this on our confocal microscope. This is a high-resolution confocal image, but when you go to really, really small structures, the diffraction limit will prevent you from seeing anything smaller than that. And so once you do STED on this exact same sample, you're able to get maybe a 5 or 7-fold improvement in resolution that in this case revealed this really cool sort of nano assembly of spectrin and inductin proteins along the axon filaments and allowing this electrical transduction. And you can use the STED microscope as you would with a confocal microscope and acquire really nice 3D images like this pollen particle, for example. So that's one super resolution technique that can give us this 40 nanometer or 30 nanometer resolution. There's another flavor called stochastic optical reconstruction microscopy that's going to work through a wide field microscope to get us down to that 20 nanometer resolution. And the way that this technique works is that you have your sample sitting under a special buffer, such that when you excite the sample with a high amount of light, the buffer will cause these molecules to switch on or off. So for example, here we have an image of cellulose fibrils that have been labeled with cyanin 5, and this is a high resolution image as far as an optical microscope goes. We're using a very high numerical aperture objective, but again, when we go down to such a small scale, the image becomes blurry because of diffraction, and our fibrils at this scale are fairly pixelated. But this sample is also sitting under a special imaging buffer, and when we excite the sample with a lot of light, initially these molecules are all going to emit at the same time, right? Because of the image buffer, the molecules will start rapidly enter this dark state and go back to an on state. And eventually, most of the molecules will have actually dark state, and only some molecules are emitting. And because only one molecule is emitting the diffraction in its space, have these isolated clear emissions that we can localize with the computer algorithm. And this process is very randomly, like a movie of 10,000 to 4,000. We have all these different molecules turning on and off, and we can localize those molecules in different frames to eventually generate 100,000 million localizations within the image that we can, at the very end, combine together to reconstruct a super resolution image that would go from something like this to something that has up to 10 times more resolution, which in this case revealed a labeling pattern along the cellulose fibers, and allow us to even see twisting within the fibers down to a sub 50 nanometer scale. So there's all sorts of different super resolution techniques, and they all have different requirements on the way that you need to prepare your sample. But regardless of which super resolution technique you use, or even if you're not using super resolution microscopy, you should always keep these concepts in mind if you want to get a nice image. And one of the important things is actually choosing the right objective and the right substrate so that you have correction for the changes of index of refraction between the sample or even just the refraction happening from the cover slip. So typically, if you want to get high resolution images, you want to use a glass cover slip with a well-defined thickness that matches the correction of the objective. You also want to use good mounting media with a high index of refraction as well, and you want to spend some time to optimize any sort of correction that you can apply within the objective or the microscope itself to correct for refraction across the sample. And what that's going to allow you to do is get very nice isotropic images in the Z direction and reduce all these other aberrations that can happen from refraction. So that's all I have for you guys today. I hope this was a very, very quick crash course on optical microscopy and the different sort of applications that you can see within these techniques. And we'll be around in the conference. Lots of different experts within the field of optical microscopy will be available with their knowledge to sort of help you meet your research goals using these techniques. So thanks again for listening, and I look forward to seeing you guys at the conference.